Good morning. I have the pleasure today to introduce Gerald Fink. And to this audience, I don't expect that Gerald Fink needs an introduction, but since I've been asked, I'm going to do it. A few great scientists can stand at the peak of their career and know they have set the path for all the important work that has happened in the community. Gerald Fink is such a scientist. He, more than anyone else, is responsible for what we now know as the awesome power of yeast genetics. This awesome power has impacted on almost all aspects of cell biology, from control of the cell cycle, which is essential to understanding cancer, to protein transport, to essential to understanding how cells function and form themselves, even to subjects as esoteric as aging. The awesome power of yeast genetics has changed everything. The evidence for this statement is his groundbreaking work in 1978, where he developed the technology to transform yeast and to target specific genes in yeast, and therefore turn yeast into one of the first organisms we can think about as a eukaryotic in approaching from a systems perspective. And this discovery changed the whole field and changed science in a very rapid and dramatic way. And for this discovery, he received the Molecular Biology Prize from the National Academy of Science. It's a prize in which 25% of the people who have received that prize have gone on to receive Nobel Prizes. It's a very significant and very illustrious prize. Gerald's discoveries did not stop in 1978. He's instrumental in our understanding of the biosynthetic pathways for many amino acids in yeast, and he's also instrumental in the later stages of his science for understanding the molecular nature of the switch between benign growth by fungal agents and invasive growth by fungal agents with his discovery in yeast. He's received many recognitions for this work. I'll just mention the 2001 George Beadle Prize from the Genetic Society and a very illustrious prize. George Beadle is one of the great founders of genetics. Gerald's also been a leader in establishing the field and leading the field of yeast molecular and cell biology. He and Fred Sherman established the yeast course at Cold Spring Harbor in 1970 and has taught that course, and then taught the course for 16 years. This is a course that has introduced a number of great scientists to the field of yeast molecular biology. Just to mention two that we are very aware of here at MIT, David Botstein took the course, and Ira Herskowitz took the course in their beginning to work in yeast biology. Gerald has also trained leading scientists in his laboratory, and he has been a leader here at MIT. He was among the co-founders of the Whitehead Institute, and then from 1990 to 2001, he was the director of the Whitehead Institute, immediately following Dave Baltimore's directorship of that institute. And he's exerted leadership across the department and across the campus in his activities as a scientist and an educator. I first met Gerald in 1971. The second year of his course. I went to Cold Spring Harbor that summer and met Gerald on Bungtown Road at Cold Spring Harbor. I've been an admirer of his work over all the intervening years and an admirer of him as a scientist and as a person. He's a person of remarkable integrity and a remarkable ability to explain his interests and his motivations and to move people in the interest of both science and education. He attended Amherst as an undergrad and then got his PhD from Yale, and he has honorary degrees from both institutions. He's a member of the National Academy of Science, the American Academy of Arts and Science, the American Philosophical Society. If there's a illustrious academic society, Gerald is a member of it. And if you want to get Gerald excited, you want to get him animated and talkative, you have to ask him about fishing. And then you'll get these long fish stories of all the very large fish he's caught and what he was doing last weekend. And he's a person who's just a joy to have as a colleague and a joy to have as a friend. And it's an honor to be able to introduce him to Jerry.
Well, I'm amazed that so many people have shown up to hear about my nativity. I don't know where to begin here. Perhaps I can tell you a little bit about where I grew up, which could give you some feeling for how I got to where I am, although I'm not sure you could trace the path very carefully. After the Second World War, my father, who I hadn't seen for much of the first 6 years of my life because he was in the Pacific as a physician, had never earned a penny since he graduated from medical school, since he was in the Army. Maybe earned a penny. And he set up his office in a small town about 15 miles south of Cold Spring Harbor on the south shore of Long Island called Freeport. A very provincial town. It was 30 miles from New York City, but it might have been several thousand miles from New York City. When I was 6 years old, one of my early memories was my mother and I walking along, and there was a cross burning on the lawn of the church, Our Lady of the Holy Redeemer. The Ku Klux Klan. She told me, sparing me some details, that they hated Catholics. So I present this as a setting for this town because you should note that David Baltimore, there's the Golden Triangle. David Baltimore grew up about 15 miles north of there in Great Neck, New York. Josh Letterberg grew 15 miles west of there in Brooklyn, New York. Okay. And Harold Varmus grew up about half a mile from my house in the same little town. Okay. So that was the Golden Triangle. Might sound like there was something interesting about this town, but it wasn't a very interesting place. Unlike Great Neck, New York, where David grew up, which is sort of the Great Gatsby, rich people lived there and so on. Freeport was a rather, mostly clam diggers, people, ne'er-do-wells of various types. And in high school, the most important people were athletes and cheerleaders. Okay. I was not immune to the cheerleaders. And so I played basketball in high school. Figured that was on to greater glory until I realized how many people were taller than I was. I should say it did give me some important experience. I played against a number of people who ended up being professionals. Okay? And I realized what the difference was, even at that stage, between a professional and a rank amateur. I could never. And I'm talking about people who were not bigger than I was. Larry Brown, who ended up in the professional basketball player and the coach of a number of NBA teams. I guarded him for 4 years in high school. But you know, it was as if I wasn't on the basketball court. I mean, you know, and people knew these guys from the time they started, and they were so much better than everybody else. Okay, and so I think it's something that stuck with me even in high school, which was that there is a real big difference between people who commit to something and have some special talent in it, because I knew I could never be like one of these fellows. And then in 1957, this world was, October 1957, was changed completely. In fact, the entire nation was changed, and my life was changed, because the Russians put Sputnik up. And for those of you who don't remember, probably too young to even, the Cold War, remember? Okay. This shocked the nation and changed this little town, which is was my view of the world, because suddenly everyone thought that it was their duty to do something to prevent the Russians from putting up something that would bomb us. And to give you a feeling for this, the Grumman and Republic Aviation, which had many engineers on Long Island, those engineers felt it was their public duty to go to the local high schools and teach calculus. And after school, I took engineering drawing, which somehow in those days was a big deal. I had terrible handwriting, and so, but we had PhDs come to our high school, and on their own dime were teaching, and the town itself put up a tax referendum, put more money into the high school, so they could, and actually the teacher a fellow who came from, Mr. Major, who came from Grumman, taught us calculus our senior year simply because he wanted to do it, and eventually he quit and started teaching high school. So I think it's difficult, if you didn't live through this, to realize how profound the Sputnik was for American science. I've often thought it would be great for biochemistry if the Russians had landed an enzyme on the moon. It definitely affected my life and my friends as well. My two closest friends, who I think had no more aspirations at that point for science than I did, although we were all interested in it, one ended up being a mathematician and the other one a physicist, and I think this definitely changed their view of things. At the same time, a friend of my father's who worked at Brookhaven Lab, named Jake Amons, he had gone to MIT. He had gone to Amherst College for his bachelor's and had gone to MIT in organic chemistry and was an organic chemist. And he was working actually on the synthesis of dihydroxyphenylalanine at the time. And in fact, it's amazing to me that we haven't made more progress because this was 1957. And I went to his lab at that time and sort of helped out in the lab. And he was married to an artist, and I think he was the only bohemian in the entire town. Okay, and so he used to have these parties that he would invite me and my girlfriend to, and they were not like the usual parties I was used to. He had a friend who was an organic chemist at the University of Utah. His friend, you can see this was not a compatible mix in the moment, because his friend was a confirmed bachelor who was an alcoholic. Okay, well, at the University of Utah, this doesn't work. Okay? And so he used to invite us to these parties at his house, and I remember opening up the refrigerator. The top shelf was all 6 packs of beer his friend brought, and then pictures of martinis. Well, you're in high school, and it's not that I drank all these things. It's more that it was sort of, it caught my fancy that there was this other thing. And the other thing that struck me was he seemed to know people outside of Freeport, okay? He knew people everywhere, you know, and this guy was doing this. And I suddenly realized that, you know, there were people who knew, you know, there was a community of some sort that was different from the local community. And that aroused my interest in science. I thought I might want to be an astronomer after Sputnik went up, but it just, I don't know why. Oh, I remember seeing Sputnik. That was the other thing. You could see it going across the sky. It was very scary, you know. I mean, in Washington, they got scared, but for kids, it was great. You could see Sputnik going across the sky. And at that point, you know, I wasn't sure what to do. Of course, everyone in our family was doctors, and my father wanted all of us to be doctors. I was immediately cured of anything like that. My father used to take me to operations at the local hospital, and I became well known amongst the nurses because I passed out. Blood pouring out of people just somehow. There's always a nurse standing there with smelling salts for me. So I wasn't really sure I would do that. But I went to, and when I applied to college, I really liked this fellow I had met and thought this was interesting. And I didn't apply to 500 schools the way kids do today. I'd applied to, actually I applied here, and to a bunch of other places. But Amherst caught my fancy. It was small, and it had ivy somehow. That, you know, that's what I thought was the right thing. And I really enjoyed the education there. And part of it is interesting because we have the core curriculum here at MIT. You know, the first 2 years, institute requirements, you don't have much choice. When I was an undergraduate at Amherst, you had no choice either. First 2 years, everybody took physics, everybody took humanities. There was no choice, zero choice. Okay. And poets had to take calculus. Okay. You know, we took courses. And actually at that time, I was quite confused as to what I wanted to do when I was a sophomore. I was taking a course with, in the Continental Novel by Professor Benjamin DeMott, and who was an extraordinary teacher. And there were many of them. And it was on prom weekend, and he, at the end of the class, he read to us from the Grand Inquisitor scene of the Brothers Karamazov, okay? In those days, the bell rang, and you went to your next class. The bell rang, everybody just sat there, stunned by this performance. And I thought, God, I'd like to do that. You know, just keep them all mesmerized. I mean, this was just amazing. Okay, so I think part of the fun of an undergraduate education like that was you really got a chance to see talented people in lots of different fields, and my classmates were very talented. By the time I got, they had an NSF grant. And then I started working in a lab. I worked on fruit fly, eye pigments, and chromatography, and separating them, and so on. And part of the program, they had people coming from universities to give talks each week. And so I suddenly was moving slowly towards graduate school rather than medical school. I actually applied to medical school. And when I went for my interview at Yale, they towed my car in New Haven, which definitely turned me against them. When I complained, they told, of course, the policeman didn't care. He said, Yale offers you an education, not a parking place. Okay. So that sort of turned me off on medical school, which I was turning attorney. But I actually published a short paper when I was an undergraduate, co-authored a short paper. And one of the people who had come through from Yale, a guy named Norman Giles, who was a well-known molecular biologist at the time, told me to come down. And I actually visited, through that program, several people, someone at Yale. I went down to Hopkins. I went to 3 or 4 places. Somehow, Yale seemed like the right place at the time, and it wasn't that far from where I was, so I didn't have to move much. And I went to Yale. And there, I'd have to say, I wasn't used to the sort of, everybody addressed people not in quite the formal way that they do in German universities, you know, Herr Doctor, Professor, and so on, but close to it. In fact, my advisor, he told me 20 years after I graduated from Yale that I could call him no longer had to call him Dr. Giles, I could call him Norman. Except for Fred Richards, who was a well-known biophysicist, biochemist at Yale at the time. He and I somehow hit it off, and he was quite helpful to me. My graduate project is really interesting because I was interested, of course, Jacobin Monod had just published this wonderful paper, and on the operon and its control. Actually, de Gaulle at the time had made it requisite for all French to publish in French. Okay, and so some of their later papers, especially the one in which they described the promoter, was published in Compte Rendu as a French publication in French. I know because I translated it. You had to pass 2 languages in graduate school at the time. German I could pass easily. I had never taken any French. But I was taking it and I thought, it was a 1-page paper on the promoter, and I thought, I'll do this, okay? And we had this wonderful, I was taking this crash course on how to do French. The teacher said, I'll make it so you, and we had people from the humanities and so on in this course, and he said, I'll make it so you'll all pass this exam if you'll promise me one thing. Never speak a word of French. I can't stand the way you people pronounce things. So, and we all passed. Anyhow, I became quite well known amongst the graduate students because I had translated this article from however poor it was, into French into English. But I was very interested in the problem of whether operons existed in eukaryotic organisms. And so I looked for an organism to work on, and yeast seemed reasonable, only because when I did a growth curve, which was my first experiment as a graduate student, all the points, when I plotted it on semi-log paper, fell on a line. And when I tried to do this with several other organisms, Neurospora, for example, they would not. And somehow that seemed to me, okay, I'm going to work on this thing. I had no knowledge. And the interesting thing is I picked the histidine pathway because at the time, I know this sounds like the Dark Ages, but it was the only pathway in which all the reactions, the sequence of reactions, were known. Okay? What I didn't know and realize at the time is, so the plan was to see if there were any operons, clusters of genes, measure their enzyme activities, et cetera, et cetera. The problem was that, showing my naivete, was none of the substrates were commercially available. Okay? And this played a big role in my career because I read all these papers in JBC, which, by the way, for those of you who only read the 3 big ones, okay, if you actually want to repeat an experiment, okay, better read a paper in JBC because you actually get the details. Okay? And I found out that all these papers came from Bruce Ames' laboratory. He was at the NIH. At the same time, I had ordered one of the substrates, PRPP, okay, which cost an extreme, for those days, $2,000. I was a starting graduate student. And my my advisor said, why don't you, figure out whether this is really what you want to do. He had some sort of locution here. And any rate, I was pretty bold. I thought, I can't do this. I read all these papers, but some of the syntheses were quite difficult. And these were all phosphoribosyl derivatives of ATP. And so I called up Bruce Ames, who was down at NIH. And he said, sure, come down. Okay. He said, come down. And I said, well, there's this PRPP problem. Okay. He said, oh, no, we're at the NIH. We can buy grams of PRPP. It doesn't matter. Okay. He said, the only thing is, you have to give us half of everything you make. That seemed like a fair deal to me. Okay. So I came down, and he and his wife put me up at their house for a week, cooked these fantastic meals. Okay. His wife is just a terrific scientist, Giovanna, and it was just great, and the lab was terrific, and it sort of was the distillation of what I thought would be a great life, which was camaraderie. He was a well-known scientist at the time, but come down, synthesize the stuff, and everybody in the lab, I had a 3-meter DAE column that we had they had a hole in the ceiling, and we had to put it up because we had to separate all these things coming out. Anyhow, and it was great. So where I was going to do a postdoc, I never even thought about a postdoc at the time. Now everybody is worrying about how their graduate students, how they're going to spend their retirement. I mean, I didn't even think about a postdoc. I was just putting one foot in front of the other. But I never even applied anywhere else. I called Bruce up and I said, I'm thinking about coming down. He said, come down, come to the lab. And so, I had 2 great years in his lab. And we've been terrific friends ever since. Graham, I think, had a similar experience, although he was more youthful than I, but Bruce is just a unusually quirky and gifted scientist, I would say. So I had a terrific time at the NIH. It was an unusual period for American science. The Vietnam War was being fought. We were not allowed to, unlike today, there was such resentment against the war that Washington was a really exciting place to be because there were enormous numbers of protests against the war. But as public employees, federal employees, we were not allowed to sign any petitions or anything. There's the Taylor Act, and it didn't let you do it. So we had to say, resides at the NIH or something like this for our. So it was a very exciting time there because many of the people who culturally, their families wanted them to be doctors. They didn't have the resolve to resist, went to medical school, but the Vietnam War offered a great opportunity because if you went into the public health service and went to NIH, you could avoid military service. And so they were known as the Yellow Berets. Okay. And the people, the list of people who were my compatriots at this time, Phil Leader, for example, Roy Vagelos, I just go down, some of the great scientists of that time were people who were basically renegades from the medical profession, went to the NIH, and so they were all people I got to know there. I don't know. Then I was looking for a job. There were many amusing aspects of that. The lab at NIH was quite extraordinary in that it was they tried to model it on the laboratory molecular biology in Cambridge at the MRC. So we had a crystallographer, David Davies, a truly extraordinary biochemist in Marty Gellert, Bruce, Bob Martin. Bob Martin is Judith Martin, Miss Manners' husband. We used to call him Mr. Manners. It was just and the whole group was headed by a guy named Gordon Tompkins, who was also a jazz clarinetist who played with some of the groups and well-known groups in Washington. And so it was a very cozy group. At one point, we were all going to move to Yale. The whole group was going to move to Yale. That was the big plan. But Gordon was, I don't know how to say, flighty. He had signed his contract, and Bruce and Gary Felsenfeld and David and Marty, and we were all going to go. It was in Time magazine that the whole group was going to move up. And then Gordon walked into the lab one day and said to me, "Do I look like a Yale professor to you?" I said, "I don't know, Gordon. I mean," he said, "Come on." I said, "Well, no." He said, "That settles it. I'm not going." I think he said this to everybody who was there, but he walked around saying these things. And I can't remember how, but I ended up at Cornell, where I had 15 fine years. It's a wonderful institution. And one of the things, I always enjoyed teaching and felt it was a part of your response. It's giving back. My father always was a fairly tough-minded fellow, and he always said, "Did you leave things better than you found them?" That was his sort of mantra. Did you leave things better than you found them? And teaching somehow is the opportunity to do that. So you have the students, they didn't know something, somehow it was simple. Actually, Gene, while I was at Cornell, long before the Whitehead, Gene had called me and asked me to come to MIT. Would I be interested, and so on. And I was a little hesitant. I didn't know the Boston scene that well, and I had heard all these horror stories about Harvard. I mean, I had lots of friends at MIT, but David Botstein can sell you anything, and so I wasn't sure whether I should believe what he said. And I have to say that eventually what decided me was talking to a number of people that MIT actually took teaching very seriously. And I have to say, one of the things that I feel really good about is the culture of teaching here, that people do take it seriously. They realize that it's not easy, I'm glad I made the decision. There were many reasons why I made the decision to move at the time, but I felt Cornell took teaching very seriously, and I felt that it wasn't, a change from that. And so I'm here. I don't know why all of you are here, but.
Since you were at Cornell, can you just briefly mention that Mark Spector story?
So, Mark Spector. How many of you know the Mark Spector story? Not too many. Okay, mostly the sages. So, when I was at Cornell, there was a big question about how signal transduction worked in cancer, and protein kinases were first discovered as being important in signal transduction and so on. And I was in the biochemistry department at that time, which was a very distinguished biochemistry department. In addition to one of the great elders of the field, Ephraim Racker, there was Quentin Gibson. I mean, there was just a whole list of really terrific scientists. Leon Heppel. These were really fine scientists. Bob Holly, who later won the Nobel Prize for tRNA, first sequence of a tRNA. And so it was a great department, and it was mostly bioenergetics, which I must say I didn't know anything about, but that's what they were interested in. And Racker had a graduate student named Mark Spector, who had a rather foreboding name. I didn't know much about him, but one day, right after we had discovered transformation, F. Racker called me down to his office and he said, I'd like you to help us clone. What Spector had claimed he had done was to have identified 4 protein kinases which were key to carcinogenesis, okay, in the RAS pathway and so on and so forth. And they actually had a symposium at Cold Spring Harbor solely devoted to that discovery. I said, F, I have no, what are you talking about? This is cloning mammalian things, you know. And it was the early days, cloning a mammalian gene. This was, you know, Wigler and Axel had just figured out how to sort of do these things in animal cells. I said, I'm the wrong person. And, you know, he said to me, he said, you don't know who I'm going to give you to do this. You know, I thought, who could it be? Arthur Kornberg? You know. He said, no, Mark Spector. I said, who's he? He said, he is the best biochemist I have ever met. Okay? He said, he's a graduate student here. Okay. Now, Racker was one of the great biochemists of his time, so he had met many great biochemists. Well, I realized, you know, something was strange. I mean, strange in the sense that there was no way I was going to try to clone all these genes. Furthermore, one of my graduate students was the girlfriend of said Mark Spector. And she was not moving along really that well. And she came to me and said, we want to, how do you label yeast with P32? I said, it had nothing to do with her project. And so I said, why do you want to do that? And she said, well, actually, Mark wants to see if these protein kinases are in yeast. Now, you have to understand the situation, right? I mean, you have a graduate student who's sort of slowly moving along, wants to do something and go in a completely different direction because her boyfriend asked her to do it. Okay. When I was a graduate student, I didn't have 5 minutes to do something else. You know, there was just something that didn't make sense. Somehow I got her out of it. This guy was, so I got to know him through her, and he was very smart. He used to come, we used to have something like the biology colloquium. Some of you graduate students may know about it. Okay, occasionally I see you there. Okay. And of course, the professors sat up front, and they usually asked the questions. But Spector, okay, always asked a question, even before the professors did. It would be something like this. It would be, you know, you said you did this, but in a paper, you know, in such and such. And of course, everybody was just wowed by the fact that not only did he know this, and so he laid this wonderful artifice of knowledge, concern. so on. And the real reason he could do it was he wasn't doing any experiments. This was all a game. Okay? He had never done any of the experiments he had claimed to have done. This was all, this case has been, how should I say it? There are even ethics courses taught around this case. Okay? And they try to make a lot out of, you know, this is a really rare event. Okay? And I can tell you, he fooled even MIT and David Baltimore, because I see people nodding here. They were going to offer him a job, right, Gene? Yes? Right out of graduate school. Okay? And Baltimore spent a week at my house in Ithaca, okay, trying to, you know, do due diligence, kick the tires. And he came away saying he's the smartest kid I've met in years. Okay? So, but it all blew up. Okay, as all these things do eventually, you know. I mean, it turned out that he had wangled his way into graduate school when they checked back in the records. Okay, he had passed a bad check for $4,000. I mean, you know, there are people who enjoy fooling other people. And in his case, let me just say, you say, why did he do this? Okay? Because he wasn't interested in the science. To talk to David Baltimore and fool David Baltimore, hey, that's cool, right? You know, and so it's the Willie Sutton. You know, do you know who Willie Sutton was? He was a bank robber. And he used to call the police, yes, he used to call the police in a city and tell them he was going to rob a bank. And he did. He was relatively successful, okay?
Because that's where the money was.
You got it. They asked him, why did you rob banks? And he said, because that's where the money was. Okay? And there are people like this, you know, who get their jollies from doing things. But that's not 99.999% of the scientists I know. You know, I've known some others like this. They're rare birds, but you know, in any field, they enjoy fooling people.
How did they finally? There at Cornell. How did they finally?
There were two people, Jeff Roberts and Fokker Fote. Okay, I can tell you how it was eventually. Jeff Roberts is one of the great biochemists. Okay, but he's got a kind of sarcastic side to him. And I asked him, because rumors were going around, I said, Jeff, what do you think of all this? He said, I don't believe a word of it. I said, why? He said, have you looked at any of the papers? I said, well, casually. He said, take a look at this. And he took a paper up. And it was an SDS gel with some bands on it. He said, I've been running SDS gels for 20 years. I cannot get bands that straight on an SDS gel. That was Jeff's. Fote, who's a virologist, actually got the gels with the bands on them. And there was supposed to be, I think it was I-125 labeled. Okay, but it wasn't the right isotope. Okay, so when they checked the gels were wrong. Okay. But you know, it was a big splash because it was an international thing. Show you another how devious this guy was. He just, I don't know why we're spending so much on it. I thought it was about me. The guy was incredible. Ed Scalnick, who's now here over at the Broad, was head of Merck, but he used to be at the NIH, worked on RAS, and he was purified to homogeneity, these protein kinases. And I've talked to him subsequently. He couldn't believe that the student could purify 4 of them, okay, to homogeneity and so on. And make antibody against them. And so he wrote to Scalnick and asked him for some antibody. And Scalnick was at NIH at the time. And Scalnick said, well, you send me some of your antibody. And he said, I'm just a graduate student, take me, I want to see if they cross-react. And so Scalnick said, okay, and sent him some antibody. And the deal was that if it cross-reacted, then he would send Scalnick back some of his, which he did. Of course, what he really did was dilute Scalnick's antibody 1 to 100 and send it back to him, because it was the perfect thing. If you're a biochemist and you make antibody, what do you want to hear? Well, my antibody is better than yours, okay? And so what he got back was something which Scalnick said, your antibody is really not as avid as my antibody, okay? Okay, but it played right into this, nonsense, so on. But again, the guy wasn't really thinking about science. He was just trying to fool people.
So, Jerry, you did some work with arabidopsis as well. How did that come into your life? What was the motivation for that?
Some of these things have a peculiar history. When I was at Cornell, I tried to get them for years to, because they had a huge agricultural college, and I tried to get them to do the molecular biology of plants rather than classical plant breeding. And I couldn't succeed. In fact, Jack Whitehead had wanted to, Susan Whitehead had gone to Cornell, and Jack had called me up. I didn't even know who he was. Somehow he heard about my interest in plants, and he wanted me to arrange a meeting with the president of Cornell, who I had never met. He wanted to give $25 million to Cornell. And somehow I couldn't arrange it, which may give you some reason to wonder how I got to be director at Whitehead, but that's another story. So when I got the opportunity here, it seemed to me that arabidopsis was in exactly the place that yeast was when I started working on it. Very few people working on it, and I sort of thought it would be, and also some of the most interesting hormone in arabidopsis, which is indole acetic acid, came from amino acids. And so I thought, And also, most of the papers in the field were, you didn't have to read. I don't like going into fields where you have to read, You spend 4 years reading before you can actually do an experiment. Here you had to read nothing, okay? Okay, just go in and see what happened. And so it was a good time to go in, and a number of my students, it was hard here at MIT because there had been no plant tradition, but I got terrific postdocs and graduate students. Chris Niogi, who was a graduate student of mine, is a professor at Berkeley now, still working in arabidopsis and doing terrific work. David Bartel's sister was a postdoc in my lab, so chair professor at Rice, and so on. I had about, really. So I thought, but then the field got, everybody said, this is great. And they all got into the field, and it somehow got harder and harder to think about doing something really novel, because there were a lot of smart people in the field. And I did have one run-in. It's hard to get funding to do plant research. The NIH, somehow plants don't contribute to our health. And the NSF, once overhead is taken out of your grant, you can basically afford half a technician. But I wrote an article for Cell, something like agricultural colleges, do they have a second chance? Okay, in which I excoriated the funding and the deans of agriculture and the Department of Defense, Department of Agriculture. all of whom told me I would never get another grant. Okay. So I was actually publicly excoriated by the Department of Agriculture for saying that they didn't know what they were doing with their money. But I got out of it mostly because I like the opening, to use a chess analogy. You know, the opening is fun. Okay, the end game, is tough. And you got to crystallize it, get the coordinates. I don't know. That wasn't my style. So I would say one of the things that I like about science is, and how many fields are there where you can make those kind of choices? Figure out what kind of person you are, what fits your, style and tempo, and then do it. There aren't a lot of fields like that, and science is one of them. And I have to say, the collegiality in science is great. No one's written the right novel or background to it. All the things about science are competition. Sure, there's competition in science. It's great, but I have never, here at MIT or at Cornell, asked for a colleague to do something or help me, give me information, and them say, oh, I wouldn't do that. You might beat me out, but all the novels, that's what's going on. Here, I always get information, reagents, and the same is true. And so I think it's a terrific field because people have a sense of the greater good. I don't mean to say there isn't competition is good. It stimulates people, otherwise you get lazy. But I don't think there are many fields where you sort of have a way of tuning your own instincts together with a group of people and trying to figure out what's good for you and what's an exciting area to move into. Yeah.
Is there any one person in particular who really influenced you to choose biology?
I think one of the reasons you go to, I don't know if it's any one person, but if you're around a lot of smart people, I liked chemistry and biology. I knew I couldn't be a physicist. I had several classmates who became physicists at Amherst, okay? And, after you've taken a few physics courses and you get to a certain point, you realize there are those people who really did only spend 25 minutes on the homework, okay? And you were laboring for 4 hours on it, and suddenly you realize, these guys actually saw it. It's like professional basketball, and so I realized I'll never be. When I was a graduate student at Yale, I took a organic chemistry lab with the two great synthetic organic chemists. One was Woodward at Harvard, and the other was a guy named von Diering, who was a big guy. So I took this lab, and I was in there with all the Yale chemistry students. And, they finished these, they knew things. I don't know, they had a sixth sense about what to do about things. And they would always finish the synthesis earlier than I did, and their yields were better. And finally, at the end of the course, which I did pretty reasonably well in, von Diering came up to me and he said, "Mr. Fink, okay, you will never be a synthetic organic chemist." It was good information. I, of course, knew it. But some people think, well, be a scientist. But, there are some things that are easy for people and some things that are hard. And some things that, these guys, Yale used to turn them out in organic chemistry. They went to work at Dow and DuPont and so on. It took 2.5 years to get a degree. I don't know. So I think one of the things that I seem to have a good sixth sense in biology. let me put it that way. I had a good sniffer for where the field was going, and sometimes when a result came up, my guys are here, so I have to be a little careful. But everybody's going this way, maybe we should go that way. And history has occasionally proven me correct.
You alluded to the fact that we both spent some time with Bruce Ames, so it indicates a certainly very unusual and very creative person. I'm just personally curious whether you see things that you picked up from being around Bruce that actually impacted the way you maybe went after science in your career.
I don't know, I might ask you the same thing. Some of it is just his verve, and there was no experiment to, he wasn't big on technical inhibition, let me put it that way. Any technique, he could figure out some simpler way to do it. He once, I brought replica plating to his laboratory I didn't invent it obviously, but he had no replica plating in the lab. I brought them in. He hated the idea that you used velveteen, okay, to do this because you had to wash them. So he said, we should use toilet paper, okay? Toilet paper is sterile. We should use toilet paper. And I remember he came in on a Saturday to the NIH, and he was trying. And then he wrote to Jacob because he claimed that the toilet paper in our toilet paper is too absorbent. He said, but the toilet paper in Paris will be perfect. He said.
When I was at Berkeley, we used some carbon paper.
Yes, that's right. Yes, that just before I left NIH. I mean, this is the sort of thing that he would spend literally weeks thinking about how to get rid of replica plating, And when I left, he said, "What about carbon paper? You guys don't know about carbon paper, but you used to type, and then they want a copy. You had a carbon sheet behind it." And he said, "The carbon sheet, I'll bet it's sterile." So he called up Union Carbide, got their technical people there, okay, and he found that it's sterile. And so then he left NIH, went to Berkeley, and he put this on Graham's head, okay, which is, there's nothing better than velveteen, by the way. But this was.
Did he finally realize that?
I think he went on. He got into HPLC. And he didn't like all sorts of aspects of all the gimmickry, and he was designing new HPLC columns because he wanted to separate urine. That was his other big thing, was to look for, he said, what is something that everybody will give up and tells you about their life? Okay. And it was urine. Okay. So, well, he was on a urine kick when you were there. Yeah, so I just saw him recently, and so I think he was again, he's very quirky, but has a very generous and broad scientific. He doesn't read any journals that I read. So, whenever you meet him, he'll tell you some absolutely obscure fact, which he then puts together, the cure for cancer from that, okay? And he reads, some weird journal you never heard of, and he found something. He's usually right. Ask Bob Weinberg. Weinberg thinks he's pretty good. I don't think you guys can stand much more of this, can you?
One last question. When did you start wearing bow ties?
Bruce Ames. So I had to Bruce, Gary Felsenfeld, and Bob Martin taught a course, because many of the yellow berets who came in did not know molecular biology, which was for them it's kind of a new field. And there was a course in continuing education at the NIH. And Bruce, who wore a bow tie, said, he was going off somewhere, I don't remember. And he said, why don't you teach it this semester? Okay. And I mean, most of these guys were, we're talking about the top level of these MDs who had gone there. So I thought I'd better look like Bruce. But I couldn't figure out how to tie one. Bruce showed me how to tie one. And ties are expensive. They used to go into the soup and so on. Bow tie, if your bow tie goes in the soup, you've probably had too much to drink.
Somebody has to ask Jerry, what's your favorite fishing story?
Favorite fishing story. Every time I go out, it's an adventure. I went out with my wife in the Florida Keys. It's not a... I went out in the Florida Keys, and I paid for a half day to go out with this guy. We caught practically nothing. Okay, so it's a tough life. It sounds like a lot of fun to go fishing, but to take people out every day and try to earn a living doing it. So he had to come back in, and just as because he had another bunch of customers, and just as we were coming in, he said, "Oh, there are 3 cobia over there." First of all, I'd never even heard of a cobia. And the second thing is, I don't know how the hell, we were going about 30 miles an hour because he was rushing to get back in to take his next customers out. And I said, "Where?" He said, "Just cast over there." I said, "Well, it's my wife's turn." He said, "Cast." So I cast it over there and handed her the rod. And one of these things hit the rod, okay? I mean, hit the bait. And she was really struggling, and the rod is like, like this. And he didn't really want that fish. I mean, he's got these customers waiting, right? An hour later, she was still wrestling with this fish. Okay? Every time the fish saw the boat, it just ripped out 100 yards of line. And this is fairly light tackle, and I kept telling her, "Don't lose the fish." And he kept saying, "Look, I don't care if you lose the fish or you don't lose the fish." So finally I said to her, "Why don't you give it to me? I'll get it in." A half hour later, I finally was a 45-pound cobia, which is beautiful, beautiful fish. But I think the moral of the story is, it's amazing how much muscle a little fish has.